visiting professor in Keio University. So 2 or 3 years ago, just before the COVID-19.
3 years.
3 years. 2019. Okay. So actually, let me introduce his career briefly. And so he had been born in India and then went to the US and had postdoc training. So Don Cleveland Lab in the Johns Hopkins University School of Medicine. At that time, I was also postdoc in department, the same department next to his room, and so we were drinking every night after experiment over, and he got independent, so junior faculty in the department of the neuropathology in Johns Hopkins University School of Medicine. So in association with Dr. Hoffman, and then he had been recruited to University of Chicago. I may have some skips.
That's okay. That's fine.
And then he is now the Thomas Royal Family Professor in Neuroscience at University of Chicago. He did sabbatical in Keio 3 years ago, and he's a well-known scientist in the field of Alzheimer's disease. And so he comes to Japan almost every year, even during the COVID-19 period. Just 3 years ago, he met Professor Kenya Honda, and so a lot of microbiome scientists here in Keio, and so today he's going to talk about the role of the gut microbiome in the pathophysiology of Alzheimer's disease. Okay, Sam, please.
Thank you very much. Thanks, Eddie. So I'm sorry I'm late. I didn't think about the timing of taking the taxi cab here and everything else, but it's nice to be back here in Japan after the COVID. I've missed being here in Japan. I used to come 2 or 3 times a year. Now it's been 2.5 years, but and I spent some time in this building as doing my sabbatical in Hide's lab. So what I want to tell you today about is some recent work from the last 4 to 5 years from our laboratory, where we're trying to look at the issue of the importance of the gut microbiome and how it may control some aspects of Alzheimer's disease pathogenesis. I thought I would first give you an overview of, let's see, slides. Right, so I'll give you an overview about the pathology and genetics, and then talk a little bit about the gut-brain axis, then and tell you about studies where we perturbed the gut microbiome with antibiotics, and then we looked at the impact on amyloidosis and neuroinflammation in the brain. And finally, and there's some very interesting findings which are sex-specific differences, and we've now engaged in a number of additional experiments to look at the role of estrogen in this particular aspect of pathology, and I'll come to that at the very end of my talk. So some of this work is published, most of it is not published, just so you know. So Alzheimer's disease described by Bavarian psychiatrist Alois Alzheimer of his first, sorry. Go back. Okay. So there was also when this lady died, who was very, very young actually in age, he then performed an autopsy and stained the brain sections of this individual, Augusty, with a classical silver stain, and what you appreciate are these 2 major pathological hallmarks: the presence of these spherical structures, which are called senile plaques, and these flame-shaped structures, which are called neurophil tangles. And we know now that these are formed in a number of neurodegenerative diseases, including Pick's disease, frontotemporal dementias, etc. It's not specific for Alzheimer's. It's the plaque that really is the signature lesion, the cardinal lesion for Alzheimer's. And one can imagine, in a cartoon-like fashion, you have intracellular accumulation of neurophil tangles, which are composed of phosphorylated forms of the microtubule-associated protein called tau, and the extracellular deposition of Aβ peptides, which I'll tell you about in a few minutes, that are surrounded by neuritic structures that are either axonal or dendritic in origin. What's not shown here, of course, is the presence of these other cells in the brain, microglia and astrocytes, which play a very important role in the neuroinflammation. I'm going to tell you more about that as I go on the talk. So the course of disease, it begins in the hippocampus, where you make new memories, thinking and planning, etc. The disease then sort of spreads from one part of the brain to another. In the mild to moderate stages of disease, language areas are significantly impaired, and in very advanced stages of disease, the entire brain is full of this pathology. So not just the hippocampus, but it's throughout the entire the cortex. And with some sparing of the motor cortex for reasons which are still not known, but it's a sort of a spreading pathology. So in gross, one could imagine what one see is the presence of these very large ventricular enlargements, thinning of the cortex, etc. The hippocampus, which would normally be the hippocampus, which is here, is not present at all in the patient with advanced Alzheimer's. So really, it's a major region for memory formation, and it disappears in people who have late stage disease. And in all cases, the presence of plaques and tangles are present throughout the entire brain, as I showed you, and they're now very good biomarkers that can detect specifically the proteins that are present in the plaques and tangles. And I'm not going to go through that work now, but suffice to say that that's really a very big area in the field is to identify biomarkers of disease. So the clinical features of disease are kind of described by this famous artist named William Oltmeier. Mullen was from New York, but spent a lot of time in London. And this is basically a self-portrait of this person who over a 20, 30-year period just had, his brain was just shrinking, it was just degenerating. And so it's basically, it features memory loss, difficulty in language calculations, behavioral alterations, psychosis, and dementia. And people die from disease, not because the brain is shrunken, but because they are usually bedridden and incontinent and die of intercurrent medical illnesses, such as pneumonia, etc. So the demographics, I'm sure you know, it's a disease that affects a large population of individuals over the age of 65 and certainly over the age of 85. So it's an age-associated condition. Given the demographics around the world and the aging population in the world, we estimate that in 2050 there will be about 80 million patients worldwide, with 50 million coming from India and China, where life expectancy has increased significantly. Extremely expensive to maintain the patients and their families. It's estimated in 2015 that it costs about $385 billion in the US alone, and there's neither a cure nor effective treatment, but there are some interesting new drugs that are available, that are becoming available and are being tested in clinical trials. So again, Alzheimer's disease, the major risk factors are, of course, age. The presence of mutations in presenilins and APP cause early onset autosomal dominant forms of disease. So they occur very early in life. They can occur in the second, third, and fourth decades of life, and they're very aggressive. And in all these cases, it has to do with the presence of the increased levels of A beta peptides, or types of A beta peptides, as the peptides that build up in the plaques. But these are very rare. There's probably about 600 to 700 families worldwide with these mutations. For the larger population of individuals with Alzheimer's disease, it's the inheritance of a number of polymorphic loci, alleles. ApoE4, I'm sure you've all heard of, is a major gene for late onset disease, but it's not all or none. It's a strong risk factor. But there's a number of additional genes, for example, BIN1, clusterin, TREM2, CD33. And what's interesting about all these genes, including ApoE, is that they're all expressed in microglial cells, so innate immune cells in the brain. And so that's going to be important when I come to the latter part of my talk. So there are these risk alleles that we think are, which play an important role in innate immune response. And we think that they're probably involved in A beta clearance. So it's the inability to clear the amyloid deposits in the brain is why we think that late onset disease is driven by these mutant alleles. The population of neurons which are vulnerable in all cases the same, the hippocampus and frontal cortex, and of course the cytopathology is in all these cases about the same. So what is A beta? A beta is a small peptide derived from a much larger precursor protein called amyloid precursor protein. A beta deposits both in cerebral vasculature and also in the parenchyma. this large precursor protein called APP. Oh, that's good. That's better. Thank you. So this large precursor protein, we still don't know after 35 years what this protein does, what its function is. Suffice to say, it's expressed in all cells throughout the body, and but does undergo this processing by these two enzymes called beta secretases, a type 1 integral membrane sprawled protease, and then this membrane tethered stub is then processed by this very unusual protease called gamma secretase, which carry out as a catalytic reaction within the lipid bilayer to release this A beta peptide. So the strongest evidence, I guess, for the role of A beta and APP in pathogenesis of Alzheimer's disease really came from studies of families with early onset Alzheimer's disease. In fact, the very first mutations were described in the APP gene, the 17 mutations here in the transmembrane domain of APP, mutation here, which actually increased the levels of A beta peptides by increasing the susceptibility of beta secretase to cleave at the site. And there's additional mutations that are found within the A beta peptide sequence that change the fibrillization characteristics of the protein. Mutations here, they have an interesting effect on A beta peptides. They change the type of A beta peptides that are generated. A beta 42 is a highly fibrillogenic, nucleation prone peptide. It's usually found about 10% in plasma and CSF, but with these mutations, the level increased to about 20 to 30% of all A beta peptides. So we think that that's what sort of drives the pathogenesis. I guess the most important study that was published in 2012 was a study that was performed in Iceland, where they looked for mutations or polymorphisms throughout the genome that protect against cognitive decline and Alzheimer's disease. And Iceland is a perfect population because it's highly homogeneous at a genetic level, and they basically sequenced the whole genome sequencing of 1,800 Icelanders, and what they find is a single mutation in the entire genome that protects against cognitive decline and Alzheimer's disease. And this mutation is actually found here. It's very close to the beta secretase site. And this mutation actually decreases the levels of A beta peptide production just about 20 to 30%. It's a very low change in the levels of A beta peptides. But that is sufficient to protect people from cognitive decline and Alzheimer's disease. So I think that was the strongest evidence, for me at least, for the importance of A beta peptides. So what does A beta peptides do? They play a very important role in synaptic transmission, and this is work that Roberto Malno and I published many years ago. We would show that these A beta peptides in some sort of aggregated form, they're called oligomeric species. They interact with postsynaptic receptors. There are many receptors that have been described. No, I don't think we actually know which are the specific receptors for A beta peptides, but what it leads to is synaptic depression. So the mechanism of internalization of these receptors is not really fully understood. What the nature of these oligomeric forms is still uncertain. Many people think these oligomers are dimers or tetramers and even a ninemer. So there are different sort of A beta peptides. It depends on who makes it and which laboratory. But what they all do is that they change synaptic transmission. They actually lead to synaptic depression. So in the early 90s, when I was heavily into the biology of APP and presenilins, Dave Borschelt and I, Dave Borschelt was a research associate in my group at Hopkins. We generated transgenic mice, and many transgenic mice have been developed, but these mice have expressed mutant forms of APP and presenilin. And what they do is they develop high levels of amyloid peptide deposits throughout the brain, throughout the cortex and the hippocampus. The one thing about these animals, just like any other, most every other transgenic mouse model of amyloidosis, is that they don't recapitulate everything that you see in human disease. So you don't see cell death, you don't see neurofibrillary tangles, you do see a considerable amount of neuroinflammation. So these models are really good for looking at neuroinflammation and for looking at a deposition. So if you look at the pathology over time, which you can do in these mice, of course, we came up with this scenario that A beta peptides that are released pre and postsynaptically influence the structure at the synapse itself, leading to synaptic pruning, so to speak, infiltration of microglia and astrocytes, which actually cause enough neuroinflammatory damage at the nerve terminals, which then subsequently leads to the cell death of the neuron. So this has been named the amyloid cascade hypothesis. That is the overproduction and enhanced aggregation of Aβ42 disrupts synapses, leading to neuroinflammation, microglial and astrocytic activation, progressive synaptic and neuritic injury, generation of tangles, etc. So this has been a sort of the ongoing model for the pathogenesis of Alzheimer's disease, and it's really the driving force for all the clinical trials that are ongoing in the field, certainly for the last 15 to 20 years. And that is really to address this issue of A-beta and the plaques, because that's what starts off the whole process. So you want to get in early when you're thinking therapeutically, and then try to prevent the rest of these events if you can really intervene very early. So I was always interested in trying to figure out how we can modify this pathology. And we published several years ago that exercise and environmental enrichment actually can largely attenuate this plaque pathology in these mouse models. But about 5 years ago, I was quite interested and keen reading a lot of literature that was emerging in the neuroscience world about the potential role of the gut microbiome in disease. And in fact, for you physicians in the audience, you recognize the name Hippocrates. So you take the Hippocratic Oath. Hippocrates back over 2,000 years ago said that all diseases begin in the gut. So that was interesting for me, by thinking about what possibly could happen. Well, it turns out that there are trillions of bacteria that are in the intestinal lumen in humans. There are as many bacterial cells in the human gut as there are total cells in the entire body. So there's a lot of cells there, and they do a lot of very interesting things. They make neurotransmitters, neuroactive peptides. They get into systemic circulation. They affect the cells that are in the border of the lumen, B cells and endocrine cells. They also have a long-term projection. This one particular cell type, these enteric neurons, have long-distance projections by the vagus nerve, right, directly into the brain. So these cells here have a huge amount of control about what is produced in the gut, whether it's tryptophan metabolites, short-chain fatty acids. These cells that are affected very close to the lumen produce cytokines, TNF-alpha, IL-1, IL-4, IL-10, many other interesting proteins, which we know can get to the brain. They do enter the brain and can have significant effects on brain function. So what is the biological basis of this? Well, the one very simple experiment that has been done now for several years is to look at germ-free mice. So these are mice that don't have any bacteria. They're completely devoid of bacteria. What happens in these germ-free animals is their brain is really messed up. There's so many changes that occur in the brain in these germ-free mice. It's unbelievable. There's increase in myelination. There's increase in synaptic and neural plasticity. The microglia are in very immature state. There's increased neurogenesis, increased dendritic growth. There's increase in blood-brain barrier permeability. All this happens when you lack bacteria. So how is that working? So bacteria have some significant control on many aspects of brain function. So is there any role for microbiota in Alzheimer's disease? Well, there are plenty of papers that have been written over the last 5 to 10 years showing that the gut microbiome in AD patients compared to healthy controls are significantly different. There's a decreased diversity, the microbial diversity and compositional distinct from between age and sex match individuals. So there is a lot of sort of biomarker data that suggests that changes in the gut microbiome might actually play some role in the pathology of Alzheimer's disease. But these kind of studies, they're fraught with obvious problems, right? So the biggest problem, of course, is that there's so many things that affect the gut microbiota composition. Genetics, epigenetics, the diet, food intake, environment, drugs, exercise or lack thereof, they all have an influence on the gut microbiota. So, for example, in the study that I showed you earlier from Wisconsin, so this is a very well-known study and heavily reported, but it was done in Wisconsin, which is a state where 99% of the people there are Caucasian. They eat the same food, they live in the same environment. So you can't compare, because they eat they eat different types of food, they live in different kinds of environment than the rest of the population. So those are the things to keep in mind. So the paper that really caught my attention was in 2015. It was a paper from Marco Prince's group, where it basically published in Nature Neuroscience, showing that the host microbiota controls the maturation and function of microglia in the CNS. So given that we knew that the vast majority of genes involved in late onset disease are expressed in microglia, I thought, well, maybe microbiota somehow control the biology of microglia. Microglia then are not able to function properly, and that leading to increased levels of amyloid deposition. So what they showed in that paper when they published this was these are single cell RNA-seq experiments of single cell microglia from SPF mice or germ-free mice, and you can see the transcriptomes are completely different. They're vastly different in the absence of bacteria. You can see that. So there must be some control of bacteria over the biology or the physiology of these microglial cells. You can even see this at the morphological level. SPF versus germ-free microglia, they are very, very different in structure, and I'll come to a few images in the next few slides. And even if you take SPF mice treated with antibiotics, they look like germ-free microglia. So we hypothesized then that the composition of the intestinal microbiome might play a role in modulating neuroinflammation that ultimately influences amyloid deposition. That was at least a hypothesis. So how do we do this? So we basically try to perturb the microbiome. So you basically give animals, so we've done this now in 2 independent lines. It's been validated in 5 different laboratories across the United States in different mouse models. And that is, you basically give a very high dose of antibiotics to animals between the age of 14 and 21 days, so they're still with their mother. And then you put them into regular drinking water until you sacrifice them when you know the pathologies occur. Or you can put them in drinking water with 1/50th the gavage dose of the antibiotics. So you get the same outcome no matter how you do it. So important thing to note here is that if you look at the total level of bacteria in the gut in these animals that are treated with antibiotics compared to vehicle, there's really no difference in the total amount of bacteria. What changes is the composition. So this is microbial composition in vehicle-treated animals. So these are just colors match different sort of bacterial species. Whether you give a 1-week antibiotic gavage, you see changes in many levels of different species, or if you give antibiotics throughout their entire life, you see very similar type of changes. So the composition of the microbiome changes. What happens to the pathology? So the striking thing in this study is that this is male. So these little dots are A-beta peptide deposits in the brain, decorated with an antibody specific for A-beta peptides. And you can see in animals treated with antibiotics, it's a significant reduction. This is just quantified here. So there's a 90% reduction in amyloid deposition when you treat animals with antibiotics. However, this only happens in male animals. You can see what happens in female animals. Nothing happens. So female animals are completely immune to this microbiota change. If you look at microglia, at this level of immunohistochemistry, you can see microglia under vehicle conditions. They have this large amoeboid-looking shape. They've ingested A-beta. You can see next to the plaque. But following antibiotic treatment, these microglia take up a very different morphology. They become what's known as homeostatic. These are what you would normally find that are in the brain. They're in surveillance mode. So there is a real change in microglial state after you do this antibiotic treatment. And one thing we can imagine is that in the healthy brain, where you have neurons surrounded by microglia and astrocytes, these microglia are in what's called an M0 homeostatic state. They're basically in surveillance mode. But following neuroinflammation, that is when they're surrounded by A-beta peptides, there's high inflammatory processes. These microglia change the state to what's called MGMD state, which is it's a disease-associated microglia. So they really look very different. They have those large amoeboid-looking structures compared to what you see in homeostatic microglia. So this is what you see in the neuroinflammation state, and of course you can also see high levels of astrocytes that are activated in the setting. So to really clarify this at a more biological, physiological level, we used a nanostring approach, basically a chip which has 550 microglial genes. We basically probe these chips with RNA from vehicle or antibody-treated animals and ask the question, what happens to gene expression? What you see here, this is now, this is nanostring panel, which is specific for microglia. So this is not a bulk RNA-seq. This is just a nanostring analysis of just microglial RNAs. What you can see here in this group of genes in male control versus antibiotics, a lot of genes go up, a lot of genes go down. Some don't change at all. But if you see in female animals, there's absolutely nothing happening. So the microglia are not being affected at all by the antibiotic perturbation. So what are the genes that are actually changing here, male that are going up and coming down? Well, it turns out that they're all focused right on TGF-beta pathway. So TGF-beta is a signaling pathway which is essential for homeostatic regulation of microglia. So what we basically are showing here is that following antibiotic treatment, microglia go from a MGND state to a homeostatic state. And in a cartoon, you could imagine that these homeostatic microglia sitting in the brain parenchyma that require TGF-beta signaling for maintenance, following the presence of amyloid plaques in these or other extracellular structures in the extracellular milieu, they start now expressing ApoE, TREM2, CLEX7A, etc. So they become these MGND type microglia. Following antibiotic treatment, what happens is now you basically reverse it. You restore this TGF-beta signaling in M0 microglia. So when you've gotten rid of the plaques, where you don't have any plaques left, these microglia go back to a sort of a resting state. So the question really is how important is the microbiome? And so we had to do a replacement experiment. It was basically we needed to find out, we had to establish causality. So in this experiment, what we've done is we've taken, so these male vehicle with antibiotics, you can see you can significant reduction in amyloid deposition, but now we feed another cohort of these animals with a fecal matter. So the fecal matter transplant from either non-transgenic animals or transgenic animals, it really doesn't matter. You can now find complete restoration of pathology. So I couldn't believe this result, because it really tells you, well, first it demonstrates causality, but that we can completely restore the pathology in animals which normally don't have any pathology. And this data is quantified here by confocal microscopy and image J type analysis, and we verified this immunocytochemical data biochemically. We just take the other half of the brain, subject to A-beta analysis, looking at insoluble levels of A-beta 40 and A-beta 42, and you can see that the same thing as we see by immunocytochemistry, that the biochemistry pretty much validates what we see by immuno. Again, in female animals, no effect of antibiotics. So having established causality, the question is, what is in that fecal matter that's really driving this? And so we basically asked the question, if you just filter a bacterial fecal matter preparation, filter out the bacteria and the very small molecules, what happens if you test that? So these are male controlled animals. This is antibiotics. You can see complete reduction in amyloid deposition. Now, when you give them FMT, you get the restoration of pathology, but this is now male antibiotics with the filtered story. So small molecules, small peptides, small anything that's present in the fecal matter fail to give you this rescue as you would see for the FMT. So there really is the bacteria themselves that are driving these phenotypes. I've told you that we've been doing these antibiotic treatments very early in life. So between day 14 and 21, the question is, what happens if you give antibiotics later in life? So at a time when they already have developed pathology. And what you see here is that this is vehicle treated animals. They have very high levels of amyloid deposition. Give antibiotic early, P14 and P21, amyloid deposits go away, as I showed you earlier. But now if you give antibiotics much later, at day 35 to 42, you don't see that. You don't see this reduction in amyloid deposition. So clearly you need to have some early intervention that happens obviously developmentally, and we think we're affecting the biology of microglia at some very, very early stage in development. These changes in the morphology of microglia, we see in control versus antibiotic treatment. You can see that the morphology of this microglia are really quite changing. It actually looks better here in this Imars type reconstruction. You can see antibiotic treated animal microglia. They look very homeostatic. Here there are these there's the MGND type microglia surrounding these plaques. With FMT, you now restore this microglial morphology surrounding the plaques. So clearly, there's something in the bacterial milieu that's having an impact on microglial biology. We look at another marker, CD68, which is a lysosomal endosomal protein, which you see at very high levels in vehicle-treated animals. So these are basically the MGMD type microglia expressing very high levels of the CD68 marker. But you can also see that the CD68 looks really funny. It doesn't, it's a lysosomal marker, so it should look like a subbody, but it doesn't look like it at all. It actually looks like a dying cell. So here are the IBA1 staining, which basically marks the identity of microglia, and this CD68 is all over the neuropil. So the question is, what is going on there? I'm going to come back to that in the next couple of slides. But again, CD68 immunoreactivity is gone with the antibiotics, so the microglia are back into a homeostatic state. They're not in this activated MGMD type state. However, FMT, the fecal matter transplant, now restores this pathology. So we went ahead and then did some RNA-seq experiments, and this is now whole brain. So this is whole cortical bulk RNA-seq. This is not looking at microglia-specific genes. This is looking at all genes in the cortex. And what you can see, there's the number of genes that are decreased in levels, the number of genes that are increased in levels in male control compared to antibiotic. And then with FMT, you can now restore much of these transcriptional signatures in these animals that are treated with FMT. Again, same thing here, where you have very high levels of increased levels of these transcripts in antibiotic-treated animals. You can now go back to this control state. In fact, if you just look at the differentially expressed genes between female control versus female antibiotics, there's only one. There's only one single DEG between female control versus antibiotic. This is what happens in male animals treated with this control animals, antibiotics, and FMT, which kind of sits right in the middle of these two. But there you can see 940 DAGs, male control versus antibiotic, and then 73 DAGs, male control versus antibiotic plus FMT. What this tells us, however, more importantly, if you just go do a GO analysis or a biological process analysis, what you see is that the number one genes that come up, microglial cell activation, antigen processing and presentation peptide, inflammatory response, synapse pruning. This is all biology of microglia. So if you do bulk RNA-seq and now dig in and say, what are the genes that are really changing? Turns out the genes that are really changing are those involved in microglial biology. So the question is, how important are microglia? So one way to do that is to deplete microglia. And so we did this with a CSF1 receptor antagonist called PLX5622. The caveat there is that this PLX analog also destroys and kills monocytes and peripheral macrophages, and also border macrophages in the brain. So it's not really specific for microglia, but in this particular experiment, we just want to ask the question: how important are microglia for these changes that we're seeing? So this is basically a control diet. With PLX, you have a slight reduction in pathology. Following antibiotics, of course, I showed you that there's a significant reduction in pathology in control diet. However, in PLX 5622 diet, it doesn't come down to this level. So that's very interesting. Same thing happens with FMT. You never get the full restoration. So in the absence of microglia, you fail to get the significant reduction in amyloid deposition that you would normally see with antibiotics, and you also fail to get full restoration of pathology that you would see with FMT. So that basically tells us that microglia are playing an essential role in driving the phenomena that we're observing. So the question is, what are microglia doing? Why are they playing an important role in amyloidosis? And this is actually a picture that was taken, that was published in 1990 by Henry Wisniewski, a very famous neuropathologist. And what he showed here is a microglial cell with amyloid coming out of it. I mean, it could be a dying microglial cell, it's a microglial cell that is now spewing out amyloid from inside. In fact, this reminds me of something that I read a couple of years ago, and that was a question about why tattoos persist. So if you ever ask the question, why do tattoos live as long as you live, unless you get rid of them, right? And the reason for that is when you get a tattoo underneath the skin, dermal macrophages come in, and they ingest the tattoo material. It's inert, it's made of plastic or whatever material they make it out of, but it's completely inert, right? So you can't degrade it. What happens is that these macrophages try to clear it, but they run out of gas, right? They run out of ATP, there's no more energy left. What happens is they die. So that's why you leave the tattoo there. Now, other dermal macrophages come in, they take it up, they try to clean it up, and they die. So it continues. So this is what we think is going on here. In fact, when I showed you the CD68 pictures of looking at these microglial cells that are surrounding the plaque, you can see large regions of extracellular CD68 positive structures, basically telling me that these microglia are dying, and thus by doing so are now releasing Aβ peptides that they've ingested. They can't degrade it. They release it into the extracellular space. There's now a number of experiments that have been published, both in models of MS, models of ischemic stroke, and Parkinson's disease, strongly suggesting that the gut microbiota are really playing a very important role in driving disease pathogenesis in those models. And so I'm going to leave you with this message that I think that the microbiome is causing a fundamental transformation of brain science. Certainly in the disease side of things, it's obviously very complicated, and obviously involves peripheral a number of peripheral cells and T cell populations that are going to play an important role, and trying to figure that out is really something that we really need to address in the future. But I think that this is basically something that was that Thomas Kuhn, a famous philosopher, stated that science is a series of peaceful interludes punctuated by intellectually violent revolutions. I don't think it's intellectually violent, but I think that nobody really considered the gut microbiome as playing a very important role, but it's a conceptual worldview that I think we're replacing by another. And that's thinking about how the gut microbiome can play an important role in disease pathogenesis. So I'll finish with acknowledging the people I've told you about, whose work I've told you about: Hemraj Dodia, Miles Minter, postdocs in my lab, who did the initial characterization of the antibody-mediated changes in the microbiome and amyloid pathology and neuroinflammation. Piali has done most of the work on the estrogen studies. These are other collaborators. Sichanda and Bob Vassar at Northwestern did the work that I told you about with in astrocytes. Oleg Butovsky at Harvard helped us with a lot of the nanostring studies that I showed you as well. The work was supported by a number of private foundations, Cure Alzheimer's Fund, Good Ventures Fund, Open Philanthropy. And if anybody's interested in working on this project in the future, I have several postdoc positions available. But I thank you for your attention.